sorry, our interest is in making as much as a high quality protein product as possible. So it's around what we do inside the cell, but also how we get it out for secretion systems, and then also look at how we can maximize the efficiency of getting the right things in and out from the cell.
Sorry, explain in mind.
Yeah, sure. Yeah. Is that better? Okay. I'm going to concentrate on two key systems that are probably the most widely used industrially in terms of making biologics, therapeutic proteins in microbial systems. One is E. coli and the yeast, and really focus on Pichia pastoris. For E. coli, focus on secretion into the periplasm of E. coli. And our aim there is that we try to exploit the relatively high cell density that we want to achieve. We don't want to go to very high cell density. Our aim is not to make buckets and buckets of biomass. It really is to get that balance right between biomass production and getting enough protein in there to give us the productivity that we're looking for. So with secretion, our strategies is to, obviously we can try and maintain the protein in the periplasm. Protein translocates across the inner membrane, accumulates in the periplasm, and that's actually very difficult to do when you push protein productivities at very, very high level. Protein initially begins to leak out into the growth medium, and ultimately all the protein that we detect, the recombinant protein, is found in the growth medium, along with some of the consequences of high-level productivity, which is cell lysis and release of membrane components. So I describe the release into the growth medium as, for E. coli, these are non-engineered host cells as stress-driven accumulation in the growth medium, and we exploit that quite widely, even though there are some downstream processing challenges. We could enhance leakage into the growth medium using leaky hosts, or there are some chemical approaches, or another approach is to try and limit the amount of protein that we get secreted or translocated, I guess not translocated, but leaked into the growth medium by trying to use more robust organisms or looking at the production conditions. In terms of E. coli and production of high-level therapeutic proteins, I would say there's probably a limited number of exemplified industrial systems, and I'm going to focus on one of the systems we've developed, something we call Paveway, as a platform for heterologous therapeutic protein production. So I'll focus on E. coli and then talk a little bit about yeast and industrial state-of-the-art, and really look at the state-of-the-art in Pichia pastoris. Again, here we exploit high cell density, slightly higher, though we can go higher with Pichia than E. coli. This is true secretion into the growth medium, though when you look at the quality of the growth medium, it's not secretion straight into a nice clean growth medium. And with Pichia, because of the way the system had been developed since the early 90s, all the industrial focus really is on use of one organism, Pichia pastoris, and really 90% of the work is done with one platform, the alcohol oxidase expression system. In terms of our approach and trying to describe how we think of the approach when designing new expression platforms or looking at what's happening in the production fermentation, and what we've found over many, many years is that many of the bottlenecks that are perceived are actually eliminated or reduced by tuning of what goes on, whether it's expression, translation, and actually translocation across the membrane. As I said, all the data I present with E. coli here is all with non-engineered wild-type E. coli. And at the most important aspect is getting the first step right, which is modulation of transcription, and then being able to modulate and being able to limit the specific productivity within the cell. That's the instantaneous productivity in a cell. If you looked at it one time and say, how fast is it churning out our particular protein? We think that instantaneous productivity is key to eliminating or reducing some of the stress responses, as well as matching the other capacities that the cell has. In matching the cell capacity, what we're trying to do is, if we're trying to express protein intracellularly and accumulate intracellular, is ensure it folds and we get soluble, but in terms of secretion, we want to ensure we don't get abnormal misfolding in the periplasm, even in the presence of the leader sequence, effective translocation, correct processing and cleavage of the N-terminus to give us the authentic N-terminus, and avoid what we consider some of the points of no return. We find that once the cell hits a particular point, no amount of change in the growth conditions, change in the media, adapting the fermentation conditions, actually pulls us back from that point of no return. So it's avoiding that point at which you can't return in terms of stress responses. And one example is plasmid stability, which is another example. By modulating, we can virtually eliminate from most proteins plasmid segregational stability, even at the intense processes and the densities and the productivities that we're running at. There are always proteins that don't fit in the rule. Our design in terms of process design is really to start right at the beginning, and it's front-ended loaded design and engineering, and really it's designing the strain with an intent that it is going to go into large-scale manufacturing. There is no other purpose for the strain, the recombinant strain whatsoever. The front end needs to be compatible with platform fermentation and induction conditions. The industry is now moving not to bespoke development at each molecule we get with and play around it for a couple of years to try and get the appropriate process and then move it into clinical development. We want to shorten that process definition and lockdown of the manufacturing process, initially going into phase 1 clinical trials for the particular product as quickly as possible. So we have a number of platform seed trains, fermentation processes, media that allow us to go straight in from strain selection straight into evaluation, and if everything is fine, the process is locked at that stage. It also means that going into manufacturing, the manufacturing team sees something similar each time. Although the protein molecule may change, one of the biggest problems in manufacturing is getting it right first time, and different processes with complex process additions, and do this at this point and do this at that point, and control must be within this window, are very difficult to control at large scale and avoid that loss of that batch. So it is to get that right first time by having a right first time approach to manufacturing development. And then for each particular protein, each protein is different, but it is to try and reduce the amount of protein target specific development that we do empirically. So you put it into a wide range of vectors, whole heap of hosts, try screen thousands of strains, pull them out, take something into fermentation, and then do the same thing again and empirically develop the fermentation platform to get you to where you want to be. Try and fix the problems right at the front end. One system doesn't suit all, and it is really to exploit a toolkit approach that we can exploit where the platform is very similar, and then we can select very quickly which one will work with the particular protein, or a few that will work. And that's in terms of hosts, promoters, control elements, and secretion leaders. And this is the way we always look at what's happening, whether it's at the cell level or what we're doing when we've got the cell in the fermenter, is that balance between what the homologous and heterologous pull is in relation to the capacities the host cell has, and some of the bad things that will happen if this side wins over this side, or this side makes so much that it then affects the quality of the host organism. The presence of foldases and capacity of foldases, translocation capacity, processing of the N-terminal methionine. With a lot of proteins in E. coli, you can get glycation, chemical glycosylation. Plasmid instability is very important. Supply of monomers, chaperones, inclusion of protein. The whole list is long. So starting right at the very beginning, our paveway system, we started at the beginning and said what we've got to really get right is control and modulation of expression itself. when the system is switched off, it really is switched off, and you could express a toxic protein and challenge the system. So the cell behaves as a wild type native organism until you're ready through the seed train, through the production fermentation, up to the point of induction. And then we want to induce expression. So we want tight control, having induced expression. What we then want, ideally, is we want modulation of expression. So not just switch it on, it's an on and off, or nearly on, or virtually on. It is on or off with modulation of expression through a range of inducer concentrations. Very simple to do during fermentation. So our key parameter for optimizing any of our processes is the inducer concentration. And I'll show you some data of why we think that is probably the most important thing to get right first. So tight control, basal expression, simple inducer to ensure that we're reducing cost of goods, not using any inducers that may be metabolized and then require further host engineering to avoid metabolism of the host. In our case, we're using IPTG. And at the point that we induce, we want to ensure that we have a homogeneous population for induction. So each cell virtually behaves the same way. It'll switch on in the same way. The rate of production in each cell is very similar. And that's to ensure two things. That we know maximizes productivity, but it also maximizes the quality of the product that we're getting. At the end of the day, what we're trying to produce are high quality proteins that are ultimately going to go into clinical trials. So trying to reduce the challenge downstream of reducing very closely related product-related impurities by ensuring that the cell makes the correct protein in each of the cells. That's modulation. So the system we use at the heart of our system is DNA loop engineered operators, LAC-I repressor, which has been modified slightly to make it the correct sequence. It's amazing how many LAC repressor sequences and databases are wrong, particularly at this end of them, the molecule where IPTG binds, promoter operator and our gene of interest. In the absence of any inducer, we get DNA loop, and this gives us a very tight switch off and control of basal expression. Now, with a lot of E. coli systems, you can switch the damn thing off, but then trying to switch it back on again is extremely difficult. We find that, okay, we can add our inducer, we get switch off, the loop is released, and we also find that the concentration of inducer in the growth medium relates to the activity at which this thing is now producing recombinant protein at any one time in the cell. As I said, one size doesn't fit all, so even with E. coli, we have a secretion toolkit. We have our vectors that are designed for high-level secretion, but here we eliminated the promoter strength. Secretion, we want to avoid something like that, high-power T7 system. The aim is not to have maximal power. It needs to be inducible and modulatable, and a library of secretion leaders. When we first started out developing our platform, we believed that a good enough secretion leader would probably work with the majority of recombinant proteins we were working with, and we focused on OMPE and PAL-B. What we've now discovered, that there are still protein products where, for unknown reasons, the secretion leader is very important in driving high-level secretion, and a good enough secretion leader is not good enough. And what we have is a platform of proprietary leaders that we've established function E. coli. They're all non-prokaryotic, plastid, humans, xenopus, xenophic, pig, buffalo. And in characterizing this, is carried out a bioinformatics study looking at the signal peptide leader database, finding signal peptides that had a pattern that looked close to a typical microbial leader, and then narrowing down from the thousands of leads down to I think it's around 20 that we actually physically tested with a range of model proteins, and then ended up with these 5 that actually work very well. They don't work with all proteins, but what we have found is that where one of the native or microbial leaders is just not efficient enough, we can use one of these eukaryotic leaders and get a very high efficient secretion. And in characterizing these, we found that what it gives us is a library of pushing proteins either through the SEC pathway or the SRP, or some of these actually drive protein through both pathways simultaneously. And in some cases that increases the amount of protein we get out by just using more of the secretion or the translocation capacity. In other cases, the SRP or SEC is more suited to the particular protein that we're trying to express. And then a library of auxiliary factors, novel foldases, and some periplasmic chaperone systems. And then our host library, they're all non-engineered E. coli, but they all have pre-optimized generic shake flask and fermenter screening and manufacturing platforms. So screen at small scale, and then within 4 weeks, we'll have 4 to 6 weeks, we can have a manufacturing platform established. So what would I consider the state of the art in secreting a recombinant non-native protein, so a human protein in E. coli, and 10 grams plus per liter is possible, and a lot of our processes are multi-gram per liter processes going in manufacturing, and some up to 10, 15 grams per liter. I've got here an example of a industrial large-scale fermentation with dry weight up at this end, the biomass, and then the fermentation time. So one of the obvious things we've done is also reduce the fermentation, increase the fermentation time by reducing the temperature. And that's part of the making things go slower during the process. So we induce and we get productivity. This is a 10 gram per liter process. And what I've calculated here is what I call the transfer rate into the growth medium, this red line. So this is what we see as the volumetric productivity, and this is the number of molecules of that recombinant protein per cell per second across that process. And you see that post-induction there's an increase and then it decreases, and the decrease does not decline with the point at which we find that the biomass starts falling apart. And the leakage does not, this leakage into the growth medium doesn't coincide with the cell also falling apart. There's a balance. There's at one point when the cell's all fine and the cells, the proteins getting out of the cell, and there's another point when it switches over, and we do find this tail off in the biomass, and the productivity then starts to crash down. So this is an antibody fragment in the growth medium. All the material found in the growth medium is correctly processed. In terms of there's no signal leader presence. We're assuming that if there is any cell lysis, any membrane-bound leader plus stuff, it goes down with the cell pellet. And only a small amount of leader plus material is successful with the cell. And that's very easy to separate because there are no, that's within the cell pellet, and we're doing the cell liquid separation. So what does that do to the quality of the harvest, that leakage into the growth medium? And I'm just comparing it here with what would consider a very clean harvest, which is a mammalian cell culture, CHO cell culture harvested with MAB titer about 1 gram per liter, no dilution, and this is the quality of the harvest. This is the same, this is an antibody fragment, E. coli fermenter harvest. Here's the product. Whole amount of other crud in the growth medium, and this one's actually had to be diluted at least 1 in 4 to actually get it to run on a gel. Inducer induction transport connects are very important. Inducible systems we're using to connect chemical inducers. And one of the things we find is that in terms of induction, it's not just simply getting the right amount in then into the fermenter. The conduction and modulation is influenced by the concentration that we think is seen by the cells in terms of the active transporters and performance. There is an impact of biomass concentration on the way the inducer is taken up, and there's some data from the literature looking at various concentrations of IPTG, different concentrations of biomass induction, and the recombinant protein produced, and the initial inducer uptake rate. And there is a relationship that the same biomass IPTG concentration, 2 different biomass levels, vastly different uptake rate of inducer. Homogeneity of induction is important, and this would be an example of what you might see with something like the ARA system, where this is the range between no ARA to various concentrations of ARA in the number of cells that are showing induction. With our system, what we find is that they're all about the same, irrespective of the inducer concentration used for induction. There's something we call induction lag, and this is about getting the inducer into into the cell. So measuring the specific productivity, this is with an intracellular protein, milligrams of target protein per gram per hour, at high inducer concentration, very rapid increase in that specific productivity, and then a decline. And as you reduce the inducer concentration, we get this peak decreasing, which is very important, which is what we want to be able to control. And then at very low inducer concentration, it looks as though we're not doing an awful lot. And if you expand that out, you actually see that even then we are getting this induction. But there is this lag of about 2 or 3 hours before you actually begin to see physically something going on. This lag is also replicated at large scale, and this is at 2 different inducer concentrations, low and sort of moderate with the dry weight and the productivity. In this case, at 0.1 millimolar, it's an almost instant reaction to tighter increase as you add the inducer here. And this, you see that actually if you look hard enough, it takes about 3 or 4 hours before you actually begin to see, and then it's not a gradual increase, it's wham, straight up and levels off. Very quickly, just go through PICIA, what I think is the state of the art. It is relatively widely used. It's not as widely used as you might expect. A lot of things were expected of PICIA when the system was developed. A lot of things were expected when the patents expired, but it hasn't got there. But there are some there are products, there are commercial products. This is a full list of therapeutic or close to therapeutic proteins that can be classified globally. 2 of which, this one here, Calbitor and Jetria, were the first to be licensed by the European authorities and the FDA, and they're actually manufactured by us, the bulk drug substance, in Billingham in the northeast of England. Its take-up has been limited quite a lot in terms of what you can do with the system. Although you get high productivity, it's possible. The specific productivity, the per cell productivity is very low, and typically it's less than 5 picogram per cell per day. There is a limited industrial toolkit. There are a few hosts that people stick with because that's all there is. It's an integrant system, copy number dependent. Range of promoters are available. This is what folks focus on in terms of inducible, and a range of constitutive promoters. Secretion leader library is very, very limited. The most widely used is this one here, MF-alpha pre-pro. And really, PICIA really is dependent on empirical induction development with respect to the host, the feeding strategy, the temperature, and the pH, and that really restricts its uptake. Though the state of the art in terms of what you can do with PICIA is probably exemplified with glyco-engineered yeast, and this is glycophy data. This is PICIA pastorius that's been humanized, 25 plus modifications, carries out human glycosylation. It's homogeneous glycosylation, it's very scalable. Glycosylation at 2 mil scale is the same at 20,000 liter scale. Other systems out there, there's a glycod, glycoswitch, and Oxirain have a system in Litica. And in terms of what it can do, this is probably the state of art where it's at. If you look at the blue bars, this is glycan structure for trastuzumab, which is the Herceptin molecule, expressed in your commercial product, and this is the glycan profile that's clinically validated. The same molecule expressed in PICIA using these engineered produces similar, but in some cases significantly different glyco patterns. The product is still biologically active, but it's not the same as the material that in the clinic has been qualified and validated. With secretion, a whole range of secretion titers from half of buggerol all the way up to tens of grams per litre, but typically industrially the number sits around here, the 1-2 grams per litre, and that's a low number. So volumetric can be high, driven by the biomass, but low specific productivity, which is a bottleneck, and significant process economics and cost of goods benefits if you can increase this picogram per cell per day by even a small percentage will give a huge value. In terms of what does the quality of the growth medium look like, it's not the clean super growth medium, protein secreted in the growth medium, the supernatants always look like this. Doesn't matter whether the titer is down at this end or the titer is up at a high end, it looks like this, and the only thing that changes is that how crappy it looks. And that is a bit of a challenge. Not as much of a challenge as with E. coli, but it's still a challenge. And some of the proteins towards the end of fermentation start to become cytoplasmic proteins. So, yeah. So in terms of bottlenecks, what's key to get right is they're getting the right modulation and tunability, and that is also a bottleneck in some cases. And it's not just the expression system tunability, it's a whole cascade from making the protein to getting it out into the growth medium. Treating, for example, transcription, translation, translocation as one activity that has to be balanced within the cell. And by doing that, you do avoid a lot of the false bottlenecks that work is done to try and get around and fix with strain engineering. With microproduction, there are still difficult to express proteins where on paper it should be fine. You don't have a lot, and it really struggles to get a high level of productivity, and not because it's a glycosylated protein. Efficient engineering of what I'm calling raw material supply with respect to the cell factory. Those are all the ins and outs, the induction and the cofactor. Always a driver in the industry for tighter, but in the therapeutics area, it's not at the expense of product quality. With E. coli, there's potential to actually fix some of the problems with periplasmic secretion by looking at high-level secretion systems that actually take protein directly into the growth medium via the periplasm. But many improvement and system challenges. And in Pichia, there are so many membranes that the protein has to get through from inside to outside. And when you look in the literature, and some of our literature, we find that there's always examples of bottlenecks been discovered here, bottlenecks been discovered here, bottlenecks been here. The only problem is it's all with a wide range of different proteins. So trying to pull that together is quite difficult. So I'll finish there.
Thank you very much.